Okay, this is your first lecture video on genetics. It covers the basics of genotype and phenotype and alleles and Punnett squares. And then there is a second video that covers probability, some different types of inheritance, and pedigrees. All right, so let's make sure we have our definitions. So the gene is the DNA sequence. And the allele is the version of that DNA sequence. So, as we see here, you can have an eye-color gene, and you might have the blue or the brown allele. So these are the alleles, these are the versions, and alleles differ by different DNA sequences, which will ultimately make different proteins. This information gives you your genotype. Your genotype is your genetic information. A trait, on the other hand, we describe that as what you see, the physical characteristics. So you might have the allele for blue and the allele for brown, but you may only see brown eyes. Your physical characteristics may be brown for eye color. And this is what we call your phenotype. So your phenotype describes what you see on an organism. The genotype is the genetic information that is creating that phenotype. And remember that you have 2 alleles for every gene because you are diploid. And we will talk about the alleles can be the same or they can be different. But the important thing is to remember is that you get 1 of each allele from mom, 1 from mom, and 1 from dad. And that's the basis of sexual reproduction and genetic inheritance in humans and many other animals and some plants. Okay. So what we're going to talk about is using this little square thing called a Punnett Square. Has to be capitalized because it's named after the scientist who figured it out. The Punnett Square is used to determine offspring possibilities. So Punnett Square is used to say, okay, we have mom and we have dad. And mom can contribute an orange or a red allele, and dad can contribute a green or a blue allele. Okay. And that each child gets 1 of their chromosomes, therefore 1 of their alleles from mom, and 1 from dad. So mom and dad are diploid, the children are diploid. The gametes are going to be haploid. But what I want you to see from this is that you can get all possibilities based on what you can inherit from your mom and what you can inherit from your dad. That is the use of a Punnett square to determine the offspring. Another term you might hear is gene locus, or the term loci is plural. And this just means the location of the gene. And it's not really important for us, but as you get into higher level genetics, it's important. And so here's chromosome 6, and here are some loci listed. And so they're like an address. So if you look at chromosome 6, it has 170 million base pairs. So 170 million GCAT base pairs. And then they list a bunch of the genes that have been mapped, or they find their location on chromosome 6. And then you get further, more detailed addresses. And just so you know, you don't have to know this for this class, but the P stands for the petite arm, or the short arm of the chromosome. And so what you can see right here, this is the centromere. So we always draw the centromere in the middle. It's not always the case. Some chromosomes have centromeres more towards the top, some have more towards the bottom. That's just a physical characteristic of the chromosome. But then we've used this to help us locate. So Q is the long arm, and the letter Q just came because it's the letter after P. And it helps us give each gene a unique address. And this is really important when you get into molecular genetics, where you're looking at maybe some different combinations of genes or how they interact on the chromosome. So, just a term for you to know. We are going to work primarily with genes that show dominant or recessive inheritance. Okay. So, here's your alleles. You got your mom's 2 chromosomes, you got your father's 2 chromosomes. Each parent will donate 1 allele. Alleles can be physically, so when we talk about phenotype, is when we talk about dominant or recessive. Okay. So dominant recessive describes how the alleles, or the proteins, really, interact. Okay. So a dominant allele, well, let me work on that. Let me give you that in the next slide. The other thing I want you to see from this slide is that Mendel, who was the father of genetics, worked with traits that only had 2 alleles of each trait, 1 dominant, 1 recessive. So he had tall plants and short plants. He had purple flowers and white flowers. This is not how most human traits, or most traits at all, are expressed. There's really not hardly any one gene, one trait. So even if we pretend and do stuff like eye color, brown dominance over blue, that's not really true. There's multiple genes that express multiple proteins that interact together to give you your eye color. But for this class, we're only going to do basic genetics. So we're going to have one gene or one trait with only 2 alleles. So I just want you to understand it's really way more complicated. Okay, here's our definition. The dominant allele is a trait that always shows up when the allele is present. So we represent dominant alleles by a capital letter. Okay. So when a dominant allele is present, you don't see the recessive, and I'll show you that in just a few minutes. Okay. The recessive allele is a trait that's covered up or masked when a dominant allele is present. So you only see the recessive trait when the organism has 2 recessive alleles. So we represent recessive alleles with a lowercase letter. And remember, one gene, one letter, and the alleles are capital or lowercase. So let's see what that means in real life. Well, not quite real life, but real life. Okay, sorry. So let's first talk about these terms homozygous and heterozygous. Okay, so if you're homozygous, that means you have the same allele. Homo in biology means same. Okay. So this describes genotype. All of this part up here. Okay. You can be homozygous dominant, or you can be homozygous recessive, where you have both of the lowercase letters as your alleles. And for homozygous, you need to define if they're homozygous dominant or homozygous recessive, because that's quite different. Hetero means different. So this means you have 2 different alleles. You don't say heterozygous dominant or heterozygous recessive because it's implied if you're heterozygous, you have one dominant and one recessive allele. And in fact, capital B lower, big B little b, and little b big B are the same, but it's always best to write the capital first for your allele pair because that helps you see that you have a dominant allele, and so you're going to have a dominant phenotype. So if we add this, I want you to see that here we're talking about phenotype now, your physical characteristic, your eye color. Okay. And when you have a dominant allele, homozygous dominant and heterozygous look the same. So a same phenotype can have a different genotype. Let's write that. A same phenotype can have a different genotype. So never assume because they show the dominant phenotype that they're homozygous dominant, because they could be heterozygous, either one of these. Homozygous recessive is going to show a different phenotype. So it shows a different phenotype from the dominant. Okay, and remember, don't go look at your parents and say, hey, I have, I don't know, brown eyes and you guys have blue eyes. What happened? You're not my parents. Okay, it's more complicated than one gene, one trait. So how do we determine which genes you could inherit from your parents? We have three ways. The law of segregation, which states two alleles of a gene will separate during meiosis and gametes will receive only one allele for each gene. Spell my words. Gametes receive one allele for each gene. Because gametes are haploid. The two alleles from your parents is diploid. Okay. So the alleles from the genes will separate and each gamete will get either this big B or this big B. Or a gamete could get this big B or this little b, right? Each gamete only gets one allele. The law of independent assortment says any chromosome, so any gene, or you could say allele, can go into any gamete. So that means all the dominant alleles don't have to go together, all the recessive alleles don't have to go together. You get any kind of combination based on the law of independent assortment. Random fertilization is our final way to get genetic diversity, and that means any egg can be fertilized by any sperm. And we look at random fertilization using a Punnett square. Okay. So let's look at these a little more closely. All right. So the law of segregation. Here are your two parents. Okay. We have a purple flower and we have a white flower. That is their phenotype. And we are told that their genotype is homozygous dominant and homozygous recessive, so that's their genotype. And you figure out the gametes. So you split the gametes. So this purple flower could produce either a big P gamete or a big P gamete. It's the same. And the same with the white flower, little p gamete or a little p gamete. And when you combine these, since they each only can produce one different one, your offspring are big P, little p, right? One from, we'll say mom, one from dad. So the appearance is purple flowers. Again, this offspring's phenotype. And look, it looks like mom, but it has a different genotype. So take a minute and think about what gametes this new purple flower could make. Right? And so this purple flower has two possible gametes, a big P or a little p, that it can pass on to its offspring. Okay? So this is the law of segregation. Each gamete gets one allele. And we only have one gene, so they each have one allele. Even though in meiosis, we're always talking about making 4 cells, this is still happening, but in genetics, we focus on the different, or the unique genotypes. So even though, yes, this flower would really produce 4 big P's, it's redundant. And so we don't actually have to write them all out. We actually could just write out the one big P. And the same here. So we're looking for unique genotypes to keep our Punnett squares as small as possible. What if you had a genotype that was big P, big S, little s? What would be the gametes? Here it gets a little more complicated because each gamete has to have one of each allele. So you could have a big P gamete with a big S gamete. Or you could have the big P gamete go with a little s, not gamete, allele. Big P allele with a big S allele, big P allele with a little s allele. You could have the little p allele going with the big S, or you could have the little p and the little s together. So you get 4 unique gametes. We'll talk about how to figure this out in a few slides from now. All right, law of independent assortment really is helpful when we're looking at multiple chromosomes. And so we have a parent, this is one parent, and the parent's phenotype is big G, big Y, little y. And I want you to make sure you understand that. Even though there's 2 G's, 2 big G chromosomes, 2 little g chromosomes, remember this is after DNA replication. So the genotype is big G, big Y, little y. And what this is trying to show you is when the chromosomes line up for meiosis, you can get any combination of chromosomes. So you could get a blue and a yellow, so as long as you have a G and a Y, or you could have the blues stay together and the yellows stay together. What's important is each gamete has one of each allele. So if you're looking at a G allele and a Y allele, every gamete has to have a G and a Y allele. And in fact, there's only 1, 2, 3, 4 unique gametes. So you can save yourself a lot of time by not writing down the redundant one. Okay, so what do we do with gamete information? How is this used to make a Punnett square to predict offspring? So our last part of genetic diversity and how you know what you inherit from your parents is random fertilization. And so what we have up here is the parent genotype. And you can see the letters are changing every time. The letters don't matter. The letters are assigned by the geneticist or by you doing the story problems. And an A gene just means it's a different gene than the B. A G gene is just a different gene than the Y. Okay, so don't get caught up in the letters. What you do is you take the parent genotype and you figure out the gametes. So across the Punnett Square, you put all the unique gametes from one parent. When these parents happen to have the same genotype, so they have the same unique gametes. This is from the other parent. And notice that the gametes are haploid, right? They only have one copy of each gene. In your Punnett Square, you combine gametes. So what I've done here is I've taken the big A and the big A and the big B and the big B. And the order you write them in, always write in allele pairs, so keep the letters, same letters together, with the capital letter showing first. So it doesn't matter who you inherit the big or the little ones from. Just keep the A's together and the B's together. So for instance, this one has two big A's, and then it has one large B from one parent and a little b from another parent. And this one has the same genotype, but they got the little b from the other parent. Okay, it doesn't matter. Keep the A's all together, keep the B's all together, and write the capital B first. It will help you when we interpret genotype to phenotype. Okay. These in the Punnett Square are possible offspring. These are now diploid. Okay. So maybe this represents egg, and these represent sperm, and now we have a new offspring that's diploid. All right, so let's make sure we understand how to figure out gametes. So, first one's easy. This guy can contribute an A, and this gene can contribute a B. So there's only one unique gamete. Again, this allele pair can contribute a big A, this allele pair could contribute a big B or a little b. So our unique gametes are big A, big B, and big A, little b. Not too hard. This one's a little bit different, right? So we could have a gamete with either a big A or a little a, and we could have the other gamete B, capital B or little b, or capital B or little b going with the little a. We want to make sure we have every combination. So our gametes would be big A, big B, big A, little b, little a, big B, little a, little b. Okay. So I might give you even more complicated ones. Maybe I give it with 5 genes. Here's the rule. To determine the number of unique gametes, you take the number of different alleles in the genotype for each gene and multiply those numbers together. So if we look up here, here we have one possibility, and here we have one possibility, and 1 times 1 is 1. There's only one unique gamete that can be formed. For this allele pair, we have one possibility. For big B, little b, we have 2 possibilities. So we multiply that, and there are only 2 unique gametes. Here, we have 2 times 2, which makes 4 unique gametes. I suggest if I ask you for the genotypes of the gametes, that you make something like this branching diagram. So for every other allele pair, you would just keep branching and branching the diagram until you split all the allele pairs and made all the possible combinations, and then you just follow your lines and you read it. Alright, so let's again look. Dominant recessive tells you the phenotype. So what we need to do is always have a definition of what the alleles stand for. So we're going to say big P stands for purple and little p stands for white. So what's the phenotype for Dad? Well, he has a dominant allele, which we know means purple. Even though he has a recessive allele, it's masked by the dominant. And Mom has the same genotype, so her phenotype is also purple. When we get the gametes for these two parents, Dad has a big P and a little p, Mom has a big P and a little p. We put Dad across the top, we put Mom across the side. Doesn't actually matter which order you do it, as long as all of one parent is on the top and all of one parent is on the side. And then we combined the alleles and we got the genotypes. Oops, that's not right. Okay. We got genotypes for all of the flowers, and then we interpreted them for phenotype. So we know big P is purple because it has a dominant purple allele. Big P, little p is also purple because it has a dominant purple allele. Little p is white. This is our homozygous recessive. And just to keep track, these guys are heterozygous, and this one is homozygous dominant. Alright, so let's work through a problem together. Alright, you have guinea pigs. The first thing I want you to always do is assign alleles to phenotype. So I have told you that big B is brown and little b is white. Second step, determine the gametes for each parent. So this parent can produce a big B and a little b gamete, because I'm doing law of segregation, and this parent can also produce a big B and a little b gamete. Okay. I personally always circle my gametes just to keep track and know that they're gametes. Next, what you're going to do is you're going to set up a Punnett square. So you're going to put your dad's genes, and this really should say gametes, right? And mom's gametes, I should have reminded you, they're still these guys. And then we combine the gametes to determine possible offspring outcome. So this is big B, this is big B, this is big B, and little b, little b. So I've just brought the gametes down to fill in each box. Now you have diploid offspring. Remember to always write the capital letter first. Doesn't matter if it comes from mom or dad. Then the last step is to interpret the phenotype. So big B, we know back here that big B equals brown. So we're going to know that this guinea pig is brown. This guinea pig also has a dominant allele, so it will be brown, and another brown one. And here we have our homozygous recessive, which we said makes a white guinea pig. So your Punnett Square information would look something like this. Now, no, you don't have to draw the guinea pigs, but they're so dang cute. I just put pictures for phenotype instead of writing it. So you want to keep track. Those are the steps. If you do those steps, you have the best chance of getting the genetics problem correct. If you skip the steps, you have a good chance of getting your genetics problems incorrect. All right, so just to summarize, we've talked about homozygous, which means two of the same alleles. And you can have, let's do the ones we have here. Homozygous dominant or homozygous recessive. Okay. Heterozygous means two different alleles for one gene. So here is the heterozygous. Let me make this correct. So we want to say homozygous recessive. Heterozygous doesn't get any further clarification because if it's heterozygous, you know you have one of each, one dominant, one recessive. Genotype are our letters. So genotype is represented by letters. Phenotype is our description. So since you will not be drawing purple and white flowers, you would write the words purple, 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 white. One more term is the term carrier. So recessive alleles are hidden when combined with a dominant allele. So we can call people that are heterozygous carriers of the recessive allele. Right? So these two parents are carriers for the albino allele. So they don't show the recessive phenotype, but they carry it and they can produce a child with a recessive phenotype. So you might see this when you're breeding your guinea pigs and you have these two brown guinea pigs and all of a sudden, whoop, you get a white little baby. You say, how'd that happen? Well, it was probably the recessive allele that both parents were carrying and doesn't show up until you're homozygous recessive. So if you see the word carrier, you know that that person is heterozygous. All right, a couple more terms. True breeding. True breeding just means, if I could spell, homozygous. And the concept is that true breeding only has one allele to pass down, so like big P, and in plants they can self-fertilize. So it would be like crossing big P, big P, and all you're ever going to get is big P. Okay. You could have this true breeding for the recessive, and if you mate whites with whites, or true breeding with true breeding, you're only going to ever get white flowers. So if you see something described as true breeding, you know it's homozygous. Now I want to talk about the definitions of monohybrid and dihybrid crosses, which are shown here. Before we look at those further, I want to let you know that there are some links to some good genetics videos that sometimes it's helpful to hear it explained by someone else. Okay, monohybrid means one gene, both parents are heterozygous. Okay, so it's shown here. We had a big T little t parent crossed with a big T little t parent. We have our gametes. We put them together in our Punnett square. This is a monohybrid cross. A dihybrid cross means two genes, both heterozygous. Well, let's look at that. This is a dihybrid cross. So both parents have two genes, the Y gene and the R gene, and both parents are heterozygous for both genes. So big Y, big R, little r. You figure out the gametes. You put them up and down the sides of your Punnett square, and keep your gametes outside of the Punnett square. The Punnett square is for the offspring. And so if you were looking at this information, you could realize that yellow is the dominant phenotype, because both parents are yellow. And for the R, you have round and wrinkled. So round, I'm going to say this is big Y. Round is the dominant phenotype. Represented by a big R. And if you looked at these offspring, you would see there's another color. So little y must represent green. And only when we have two little y allele pairs, homozygous recessive, do we see the green. And R, you only see these wrinkly P's when you have homozygous recessive for R. So R represents wrinkled. So this Punnett square is showing you both genotype and phenotype. Yeah, so let's take it a little harder. Let's do a trihybrid cross. Let's cross the S. Oops. Y, and A. So again, 3 genes, everything is heterozygous. So if you're looking at this to figure out the unique gametes, you know it's 2 times 2 times 2, so you have to figure out 8. And it's the same over here because they have the same genotype. And again, a way to figure this out is the branch method. You can do it sideways, you can do it up and down, but what you do is you first split one allele pair, and then you pair them up with each option from the second allele pair, and then you pair those up with each option from the third allele pair. And so if I read down my line, I would see that I have a S, oops, I don't want it, hold on. I have SYA in one gamete, and if I follow this line down this way, I get SYA, S little y big A, S little y little a, and you keep going. Oops. S, Y, little a, S, little y. I'm getting small. This is what I tell you. If you have 2 letters that look the same, make sure you make them look different, or you're going to get messed up because your letters don't always stay large and small. So there are my 8 unique gametes. For one parent, it would be the same for this parent. We put them on a Punnett square and you'd get this lovely thing. Yeah? An 8 by 8 Punnett square. Don't worry, I won't do that to you. But then you would be interpreting phenotypes based on 3 different genes. It's really fun. Okay. Phenotypic ratios to recognize. So this is kind of like shortcuts. So if you have a monohybrid cross, okay, and we're talking phenotypes, you're always going to have a 3-to-1 dominant phenotype to recessive phenotype. Okay. So you had parents that were big P, little b, P crossed by big P, little p, a monohybrid cross, and when you look at phenotypes, you have 3 purple, 1 white. 3 of the dominant phenotype, 1 of the recessive. Now the genotype is different. The genotypic ratio, and this is the phenotypic ratio, and it's just done by counting what you have in your Punnett square. But if you memorize some of these, there might be problems that you can apply this shortcut to. The genotypic ratio would be 1 to 2 to 1. Big P to big P, we have 1. We have 2 that are heterozygous, and we have 1 that's homozygous recessive. And for me, you have to always define what your ratios are. So don't just write a 1 to 2 to 1 or a 3 to 1. If you're writing out a problem, tell me what those numbers represent. Monohybrid cross, pretty easy. Dihybrid cross is a little more complicated but it always gives a 9:3 to 3:1 phenotypic ratio. And what that 9:3 to 3:1 means is that you're going to have 9 that show both dominant traits. Both dominant. Okay, so if you count it up, you have 1, 2, 3, 4, 5, 6, 7, 8, 9 yellow and round. The next part of your ratio says 3 of those 16 are going to be showing 1 dominant and 1 recessive trait. So in fact, we have 3 yellow wrinkled. Yellow is the dominant, wrinkled is the recessive. The next 3 says you have just the opposite, the recessive trait and the dominant trait. So now we have 3 that are green, but they're round, right? So they have the recessive color, but they have the dominant shape. And finally, the 1 stands for you're going to have 1 out of that 16 that has shows both recessive traits. So another way we can write the general genotype is we write this as big Y something, big R something. Big Y something, little r, little r. I'm going to write it down below. Let's see. Little y, big R something, and then our last one is little y, little r, little r. And those blanks mean it could be the dominant or the recessive, right? So if you look here, big Y, big R, big R has the same phenotype as big Y, big R, little r. Okay, so again, don't assume you know what this gene pair is. This is the only one you know for sure. If you wanted to do the genotypic ratio, which you do not need to memorize, and I say memorize the 9:3 to 3:1 because it will help you go faster on some problems, but if you don't remember it, you can always work it out in a Punnett Square. So all the unique genotypes have a 1 to 2 to 1 to 2 to 4 to 1 to 2 to 1 to 2 ratio. And you could go through and figure out where all these unique genotypes are, right? So we have 1, big Y, big R, big R. But we have 2, big Y, big R, little r. And I'm not going through all those. You don't need to memorize that number. I can't remember that number. But the 9:3 to 3:1 ratio can help you do some problems faster. All right, we got 3 more slides. How to name generations. So sometimes they will talk about the P, the F1, or the F2 generation. The P generation is the parents. And you see these are true breeding, so you know that they are homozygous. So one parent is always going to be homozygous dominant. One parent is going to be homozygous recessive. The F1 generation is what comes from those parents. So in this case, the F1 is going to be big P, little p. The F2 generation, so F1 is from parent mating. The F2 generation, by definition, is the mating of 2 F1 siblings. Yeah, I know. We don't do this in human genetics, right? But plants, when we're breeding sometimes mice for research, might breed two of the siblings and come up with the F2 generation. So just to understand these definitions, we're going to come back to this idea. I want to reinforce the question, can you tell genotype from phenotype? And the answer is not always. All right, we did that monohybrid cross and we got a 3-to-1 phenotype typic ratio, but a 1 to 2 to 1 genotypic ratio. So if you just knew all these flowers were purple, you wouldn't know which ones are carriers. So we have one more technique in genetics, and if you want to figure out the genotype of a dominant, or let's say of an organism expressing a dominant phenotype, you do something called a test cross. So let's look at a test cross. A test cross, you take an organism of a dominant phenotype, unknown genotype. So it could be this or this. And you always cross it to a homozygous recessive. Because we know this phenotype, little p, little p. And when you do this, because it's homozygous recessive, it doesn't mask any of the alleles from the parent. So they're going to show up in your offspring. So if you just look at this image, they're showing you if the parent was homozygous dominant. And we did the test cross, you would get all purple flowers because that dominant allele dominated all of the homozygous recessive. So results, all offspring are purple. But if that dominant purple flower was really, dominant phenotype purple flower was really heterozygous, when you cross it with a recessive, homozygous recessive, you get half the offspring being purple and half the offspring being white. So you have unmasked the dominant parental alleles by doing a test cross. So understand what a test cross is, understand how to use it, and understand that the different outcomes of the cross are dependent on the genotype that we didn't know. So you will be working problems with these concepts. Let's go through the outcomes and make sure we've talked about everything. All right, so we've talked about labeling generations and especially what that F2 generation means. It means an F1 crossed with an F1. We've talked about the difference between a gene and allele, and we've talked about the locus being the location. We've talked about dominant and recessive traits, dominant being the one that shows up whenever it's present, recessive not showing up unless it's homozygous recessive. We've talked about phenotype being your physical characteristics, genotype being your genetics. Your characteristic or your trait is really talking about phenotype. What do we see? We've talked about allele pairs can be homozygous dominant, homozygous recessive, or heterozygous. We've said that true breeding means homozygous, and that a carrier means heterozygous. We've worked on relating genotype to phenotype and going both ways, determining phenotype from genotype and genotype to phenotype. We've used a Punnett square to determine ratios or numbers of genotype and phenotype. So a ratio is basically when you count up the numbers of each different thing. You put these little dots between them. And this might be blue to green to brown. So you're just counting offspring. And you can count offspring phenotypes and get a phenotypic ratio, or you can count the genotypes and get the genotypic ratio. Right? And these are for the different unique phenotypes or genotypes. We've talked about the law of segregation and how that means that each gamete has to get only one allele for each gene. We've talked about independent assortment, which means that any chromosome can go into any gamete as long as you have one of each. We've described a monohybrid cross. And we've talked about the 3-to-1 phenotypic ratio. We've talked about a dihybrid cross. And the 9-to-3 to 3-to-1 phenotypic ratio. Determine parental genotype from frequencies of F1. So this is going backwards. We'll do some practice problems with this. Determine the genotype of gametes of a parent given a genotype involving 2 or 3 genes. So this was the big P, big S, little s, big A, little a, right? So you could figure out all the possible 2 times 2 times 8 different gametes. So once you feel comfortable with the terminology, go practice genetics problems. Remember, there's one more video that will talk about probability, some other types of dominant inheritance, and pedigrees. All right.